Did you know that poop transplants are a real thing? And that if you're healthy enough, that you can be paid to be a super pooper? I'm Amanda Thomas, and you're listening to A Scientist Walks Into a Bar, a podcast that brings you presentations by real scientists recorded at events called Science on Tap. In this episode, Dr. Andrea McBeth will be talking about all the bacteria, fungi, and viruses that live in a healthy person's guts. She runs Flora Medicine here in Portland, Oregon, and one of the things she does is treat people who are suffering from an infection called C-Diff. That infection can be super scary because it's often resistant to antibiotics and can lead to death. However, poop transplants, officially known as fecal microbiota transplants, are 96% effective against recurrent C. diff infections. So in this case, poop saves lives. We'll include some of Andrea's images from her presentation on our website, and we'll include a link to our website in the episode notes. Also, you may already know that we do trivia at the beginning of our events, but the audio would be boring for a podcast, so we leave it out. However, Andrea mentions trivia a few times during the talk, so I wanted you to know what she was talking about. Finally, if you like what you hear, please let us know. You can find us on Facebook, Twitter, and Instagram at SciBar Podcast. And now it's time for some scoop on poop.
Thank you so much for being here. So if I asked you to eat poop, would you do it? What if I told you it could save your life? Okay, so many of the patients I work with answer yes, and we work with a therapy called fecal microbiota transplant. So that is more or less literally what it sounds like. We take the healthy stool of a healthy screen donor, human, magical human, and we deliver it to the colon of a sick patient. And in the case of my patients, they have infectious diarrhea, specifically C-Diff, which we'll get into about what that is. But it's not a new treatment, like we mentioned. It has been around for a really long time. The first reference was in 4th Chinese literature. Golden soup was used for diarrheal disease. It fell out of favor later as we discovered that bacteria cause disease. So you think of germ theory and Koch's postulate, and as we were able to first see bacteria with the invention of the microscope with Van Leeuwenhoek, and then later Louis Pasteur and pasteurization and Koch's postulate, we learned that infectious bacteria cause disease. We stayed way away from poop. Turns out that was where a lot of disease was coming from, but we've come full circle. And we now have antibiotic-resistant pathogens that after we had the advent of antibiotics, we didn't really have to worry. We could just take those, kill these infectious bacteria, and move on with our lives. It was a really important part of health and medicine evolution in our timeline of human history. But now we have things like Clostridium Difficile, or Clostridium Difficile as it's been renamed. So if you are a healthcare provider, your pulse has probably quickened seeing this little guy. It is a very nasty diarrhea-inducing infection that is causing a lot of problems in the US and global healthcare system. In fact, in 2011, which is our most recent epidemiology, it was estimated that over 500,000 people contracted Clostridium Difficile infection. And of those, in the ballpark of 15 to 30,000 people died, which is a lot. The problem is that C. diff, like other things that can happen, especially in elderly, is a tipping point towards other problems. So you can end up with sepsis and die because you have diarrheal disease and it weakens your system. It's no longer responding to a lot of the antibiotics we have. It's kind of like we're out of the cabinet. We went to the bathroom, opened the cabinet of antibiotics, like a couple of other antibiotic-resistant pathogens like MRSA and things like that. And C. diff says, I'm here. I'm gonna keep living here. You can't get me out. Luckily, fecal transplant is something that does work. And I know that sounds crazy, but we're gonna get into why. And it has a lot to do with the ecology of the human microbiome. So to take a step back, I skimmed over this slide, but in my clinical setting, we give people frozen oral poop pills Think of it as a super probiotic, if you will. And with 10 capsules over 3 days, their C. diff goes away. Their antibiotic-resistant C. diff goes away. So where does this come from? Well, we have very magical unicorn super poopers. And you might ask me, where do I find them? Thank you, Portland, Oregon. I know we're in Vancouver, but our donors are in Portland. That's where my clinic is. And they have a lot of really unique aspects besides meeting all the screening requirements for infectious disease. They have a lot of the things that we know impact the health of that 4-pound microbiota organ we were talking about in the intro. So you might be looking at this image. It's of a happy human that pooped today, and they were vaginally born and breastfed, and they have happy bowel movements now. They've really never taken any antibiotics or pharmaceutical medications, and they've really never been sick. And this is because a lot of these things we know impacts the microbiome, and we want to give our sick patients the healthiest representation we possibly can of the microbiome. There's still a lot we don't know about why it works so well, but we do know that the donors need to be healthy and they need to be very rigorously screened. And we'll talk a little bit about that too, because like I said, with germ theory and understanding that poop, we are afraid of poop for a reason. It is the main mode of fecal oral infection. So pathogenic bacteria use that method because we can drink and get exposed to fecal material and get things like cholera and infectious disease. So it's important that the donors are screened for all those bad infections, but that we also maintain all the healthy things. So once we've found the poop, we take it into the lab. It's pretty straightforward. We use sterile techniques. We literally put it in a blender, which I know sounds gross, but at this point, it doesn't really matter to me anymore. We process it, and we pellet it. So we turn it into a really super probiotic. We take all the other stuff out of it, and what we end up with is those good guys that we need to go in and fight the C. diff infection. So we have patients who are having 3 to 30 watery diarrheas a day. They're really, really, really sick. And this can happen to not just elderly, people over 65 and 75. I had a 92-year-old lovely patient recently, but I also have 32-year-old young women come in that just happen to have antibiotics, and that creates an opportunity for this infectious pathogen to come in, and then they get C. diff, and they don't respond to antibiotics, and they're sick for months and months and months, and then they take the first 10 capsules, and within 24 to 48 hours, they're having normal stools again. Their diarrhea goes away. It's pretty amazing. I really like working with the therapy. There's been tons of clinical data on it. It's roughly 90% effective. There was a clinical trial that compared different delivery modes, specifically swallowing the poop pills with delivery via colonoscopy, which is also commonly done, and it was found that they were both equally effective at 96.2%. There's other larger studies that have been done that show they're all around 90%. Nasogastric and upper sometimes doesn't work quite as well as lower, but the capsules, especially in repeated dosing, seem to work really well. There is obviously a concern about safety because I mentioned the aspect of having fecal oral transmission of infectious disease, but overall, people seem to respond really well. The adverse events is what we call, side effects, is bloating, sometimes some transient diarrhea. In the roughly 50,000 FMTs that have been done by the large national nonprofit that does a lot of this, they're called Open Biome. Only one person has gotten into trouble, has passed away, and that particular thing happened recently. And so I mention it because some of you might be saying, this sounds really scary. Eating poop sounds bad to me. There's got to be a reason. And that's fair, but of all these people that have done fecal transplant to cure their potentially otherwise deadly C. diff infection, for the most part, it's been really safe. The one person who did pass away was immunocompromised, and their particular FMT wasn't screened for a drug-resistant pathogen. So I always come to this point, well, specifically this point, where I start to think about, well, if it's 90% effective, this is a disease that's not responding to anything else, why isn't it more widely available? Why isn't this something that, if you work in a hospital, most people with C-Diff that don't respond to the antibiotics get access to? And that is a problem in healthcare in general. And this slide, I think, really is a good representation of part of the challenge of developing a safe drug in the healthcare system under the FDA regulations. It's really expensive. You have to spend a lot of money, and it takes 10 plus years, so roughly $2.6 billion, to get a drug from start to finish. And so there's an argument that's made by different policymakers and things that fecal transplant could be regulated like we regulate blood. And I think that's a pretty good, there's arguments for that I think are pretty reasonable. We've been trying to synthesize blood for many, many decades, and we haven't been able to. It's really complex, but it does come with inherent risk. And the same thing with fecal transplant. So there is risk of theoretical infections we don't know exist yet, but there is a call that it could be regulated like we do in blood banks, specifically in Scandinavian countries. They're looking at integrating fecal transplant into the blood bank systems they already have. Fecal transplant is considered one of the standard treatments for antibiotic-resistant C. diff infections now. So we're starting to think about how we're going to matriculate this in. It's a relatively new thing. It's in a unique space in the medical system in that it is a discretionary enforcement, which means it's not really approved, but we know it cures C. diff and those people can die otherwise, so it's really important they have access to it. So we are in a little bit of a limbo until a drug can be approved that's equivalent and can replace it, or the regulation changes. So I might have slowed down the excitement of the poop, but we're gonna get back to all those jokes and fun things. And I think I start with talking about fecal transplant, one, because it's amazing and fascinating to watch my patients get better. It's really awesome. I also think it's fascinating. And I'm always come back to this question of why does eating poop cure diarrheal disease? That is crazy. Well, it turns out it's not actually that crazy if we dive into the fun part of this talk, which I think is the microbial ecology. So in the intro, the questions were about the human microbiome. And the human microbiome, what we know about it, microbiota, it's made out of bacteria, viruses, fungi. They live in and on us. 4 pounds roughly, the estimate is 1-to-1 cells, bacterial cells to human cells, and 100-to-1 bacterial genes to human genes. But I like to break it down a little more simple. We are really a donut. And we're gonna go on a journey of the human body as a donut, of the GI system. And so if you think of our skin surface, the surface of our lungs, our GI tract, mouth, to the end, as the outside of this donut, I want you to, we're gonna close our eyes, pretend we're the Magic School Bus, we're gonna take a sip of beer if you have it, or just imagine you're about to eat a bite of a donut, and you travel into the mouth, you're going down the trachea, you're going down into the stomach, your stomach empties into your small intestine, which is really, really long, wraps around, all the way down into your colon. You wave to your appendix as you pass it, if you have one still. And then you come out the end. So that is the inside part of your GI system. And then our skin surface, vaginal surface, lung surface, mouth, other parts of the external part of our surface are also epithelium and are covered with this ecosystem. And so now people are often like, why didn't I know this? We are covered with bacteria, viruses, and fungi, and we need them. We wouldn't survive without them. And most of that 4 pounds of bacteria, where the sprinkles are, lives in this part of the colon, which is the large intestine, at the end. And that is what makes our poop. If you think of the poop as like the compost of our ecosystem of the colon. So why didn't we know this was there? Why are we so afraid of bacteria if we're covered in it? If we have 4 pounds of it, why should we care about dropping our food on the ground? Well, part of it is because we didn't know they were there. I mean, we really didn't appreciate this until a decade or 2 ago. We've had microbiology for a long time, but we could only grow things on petri dishes that could survive in oxygen. And a lot of the stuff in our gut doesn't like oxygen. And so I like to think of it as we were in a room that we thought was the whole world, and we were hanging out in this theater, having a great old time, looking at like the bugs on the ground, exploring our universe that was the theater. And somebody walked over and opened a window, and there's a whole nother world there we never even knew existed. It's microscopic, and that's part of the problem. And the other problem is we couldn't grow it in traditional microbiology techniques. So really, until the advent of a sequencing technology, which is the window to this new world, we didn't know this diversity of bacteria were in our colon. We didn't know what they were doing. We couldn't see them change. Our microbiome shifts 36 hours with a diet change, the profile of our microbiome. But we couldn't look at that before we had DNA sequencing. So sequencing is a way to take a snapshot of who's there. It doesn't matter if they're alive or dead, but it gives us like the script of what is represented by their DNA. And so it's a really cool technology. I don't know, it's Science on Tap, so I'm sure you guys remember the Human Genome Project. It was a really big deal when we were starting to map the human genome. And then, take a step forward, we started mapping the human microbiome and sequencing that. And this is a graph of Moore's law. You can't really see that. But Moore's law of technology shows that the cost per genome sequencing got much, much cheaper. So all of a sudden we could take those pictures like really cheaply and look at what's happening in the microbiome. So this is where I get to all my scientific celebrity crushes. I'm sure you guys have some. My sister's really trendy. She lives in LA. She loves the Kardashians. I love Rob Knight. We're different people. My mom's here. She can appreciate that. He was really foundational in taking all of this data we collected with sequencing of our poop in different places on our skin and armpits and nose and all kinds of places, and data crunched it into a tangible way to appreciate what was happening. And so this is really relatively recent information. It really hasn't been translated into conventional medicine because it's just happening now. I love these scientists because they are brilliant and doing all this cool work, and then they write these books that are so cute, like Eat a Vegetable, Dirt is Good. And you're like, wow, that speaks to me. Maria Dominguez Bello and Martin Blazer are 2 other scientists I really love. They have done a lot of work in trying to save the diversity of ecosystems in the microbiome around the world. So they've traveled around the world and they're collecting poop samples from people in remote tribes in the Amazon, because what is happening with climate change is happening in our microbial ecosystem. So in the same way on a macroscopic level, we're seeing mass extinction of insects. We have mass extinctions in the western gut of our microbial ecosystem. And it's a problem. And we'll get to more of like what's happening because of that. But these 2 scientists in particular are really raising the alarm and doing some cool work. Maria Dominguez Bello did a study where she swabbed babies that were born via C-section with the vaginal fluid. It's called seeding, to see that the difference is because we know babies born via C-section have higher risk for asthma and atopic disease, and they also have a different microbial profile. So their microbiome looks more like the skin of the doctor than the vagina of the mother, because that's who they touch first. And so she designed a study and did a study where what happens if we put the mom's vagina bacteria on the nose and mouth instead of the doctor's skin first. And so she's really cool too. All right, this is my last celebrity crush microbiologists. The Sonnenbergs are a group at Stanford who are microbiologists, and they have done a lot of foundational work in what's happening with probiotics and why fiber is so important, and we'll talk a lot more because I love vegetables. It's like my favorite part of this. You can eat poop, but you could also just eat vegetables. And they wrote another book very similar about why feeding kids vegetables is so important, which I thought was very cute. So we had this revolution happening with sequencing technology. We sequenced the human genome, and then we started sequencing the American gut, or the American Gut Project. And we really, like when we sequenced the human genome, we were really excited to be like, okay, this is it. We're gonna solve all the problems. We're gonna figure out what all the diseases are caused by. It's gonna be this one bacteria, and then we're gonna have all the answers. And it's never that simple. It just isn't. So it turns out there isn't one healthy microbiome. Since the American Gut Project started, there's been thousands of people's microbiomes, healthy and sick, sequenced. There's tons of data out there, and it's more complicated. And this is where, when I'm at a microbiome conference or going to listen to lectures about this topic, I always give a little cheer when the ecologist comes up. Any ecologists in the audience? Woo, you're my favorite. Because it turns out it's not linear, and it's harder to look at the microbiome from a medical perspective, linear medical perspective, because it's really ecosystem biology. And just like there isn't one healthy ecosystem, we can have a healthy coral reef or a healthy rainforest or a healthy tundra, but they look really different. The keystone species in a tundra isn't going to be the same as the keystone species in the coral reef. But if there's diversity and the ecosystem is robust, they can still be healthy. They just look a lot different. And that's kind of what we see with the human microbiome, is that different healthy people can have different profiles that may look really different based on just the species that are there, but they're functioning correctly in this human interface. Another aspect of ecosystem biology that's really important is this concept of keystone species. And recently a scientist presented at a conference and talked a bunch about guilds, which I think it's such a cool word. I mean, I'm a big fan of historical fantasy, so guilds made a lot of sense to me when we started talking about groups of organisms that perform tasks that are important to the ecosystem. So we have keystone species who, when we lose them, like the gray wolf in Yellowstone, the rivers change the way they flow. So they're relatively low in their mass representation, or the beavers, but their impact on the ecosystem is profound. And then we also have guilds. And in the case of bacteria in our gut, there's groups of bacteria that all perform tasks, and there's a bunch of different species that do it, that break down certain kinds of fiber, for example. And it's not that you can lose one, or if you just have one, that's the answer. But if you lose the group that does that task, you're out of luck. So if you lose all the butyrate producing bacteria, you can't digest a certain kind of fiber. Just like if you don't have the right kind of trees in the forest and you lose your top story, you're not going to have a healthy ecosystem. So there's two concepts that I think are really important as we start to talk about what are the repercussions for this very real loss of ecosystem health that we're talking about in our gut. Well, we have a chronic disease epidemic in America and in the world, but it's pretty profound in the United States. 6 in 10 adults have a chronic disease, and 4 in 10 adults have 2 or more. So when I say chronic disease, the difference between a chronic disease and an infectious disease is an infectious disease is caused by a pathogen like C-Diff. A chronic disease is things like heart disease or autoimmune disease or diabetes, things that aren't caused by a specific pathogen per se, but more complex set of things that happen over time. So why is this happening? Why do we have this chronic disease epidemic, and what does that have to do with the poop pills, the magic poop pills? Well, it turns out that diversity is one of the only things that we found were important, we being the collective science community, which I'm excited that many of you are probably a part of. There wasn't one species everybody needs, there wasn't a clear-cut profile, but one of the things that we did see was that loss of microbial diversity, which is represented in this chart, and not like the best handwriting. I don't have the best handwriting of highlighting, but you can see here this highlighting row that I did is loss of microbial diversity and correlation, which isn't causation, but is important when we're starting to unwind what's going on here with these chronic diseases and other issues. We know the microbiome is important. We're starting to appreciate that there's a phase in early childhood, like preconception to 1 to 3 years, where the human microbiome is playing a really important role in our immune system for the rest of our lives and our risk factors for those chronic diseases. So things like C-section fall under this development window. Exposure to the right kind of good dirt, which is why Rob Knight wrote that book. We also know that the microbiome plays an important role, the diversity plays an important role of aging. And there's data that shows our chronologic age can differ from our physiologic age. So like you can feel like a 90-year-old, you can chronologically be 90 but feel like a 60-year-old. And those people tend to have the microbial diversity of a 60-year-old and vice versa. So if you are 60 and you don't feel very good, you might have the microbial diversity of somebody with a much older chronology. But we really are in the exploration phase. And I get really excited about this because I used to work in molecular biology. I stared at a lot of cells. And I always thought of myself as like a space traveler, as like in Star Trek, but in a microscope. I don't know if you guys have ever felt that way. And so when I thought about exploring the cellular mechanisms of cancer cell communication, which is I did before I became a naturopath, it felt a lot like me. I was exploring the outer space. And we're kind of in that phase with the microbiome. I love the microbiome because it is a bridge to all of the common sense, foundational health, lifestyle things that I love, and it gives me a mechanism of action for why things like taking a deep breath and eating vegetables can be so impactful on human health. But I really want to know what's going on. How are they communicating? How are they communicating with us? How are they communicating with each other, meaning the bacteria? And how can I translate that to the very sick people I see on a regular day-to-day basis? So before we get too far, I love talking about this to people, and I don't always talk to Science on Tap. I talk to other groups, and I always think it's important to point out that clinical is different than preclinical data. And so with the microbiome, there's a lot of information about mice, and mice aren't humans, so we can use that information, but if something's true in mice, it doesn't mean it's true in humans, so it's my little caveat. There's also this website that I love called Spurious Correlation. So although correlation is useful, it does not mean causation. And this website is a collection of hundreds of graphs of correlations that are hilarious. So the age of Miss America does not cause murders by steam vapor and hot objects. But man, if you were just looking at that graph, you would be really impressed with that causation, the correlation. So we have a lot of cool science happening in the microbiome space. There are mice that don't have any bacteria, and then you can introduce bacteria into them. They're called germ-free or notobiotic mice. And so that gives us a really great way to control variables and look at what different kinds of bacteria are doing. What happens to the brain development when you don't have bacteria? What happens to caloric intake versus weight gain when you don't have bacteria? It turns out it's profound. So there's a lot of cool stuff happening there. Fecal Transplant is also giving us a lot of really fascinating data because we're taking this like theoretical ecosystem model and we're transplanting it and we're seeing what happens. And so there's really cool stuff happening where you can transplant obesity in mice. It doesn't work quite so well in humans, but a fecal transplant of an obese mouse to a lean mouse can make that lean mouse obese with all variables controlled. Which is crazy. I mean, I always get excited about this, but it's pretty nuts. We also know that the microbiome is really important for the immune function, and one of the examples that I get really excited to talk about is this set of studies that was done looking at cancer treatment. So people have cancer, or these mice have cancer, and they are treated with a chemotherapy agent and it doesn't work. PD-1 checkpoint inhibitors, which are a specific kind of immunotherapy. But if you give that person or that mouse a fecal transplant and then give them that chemo again, it works, which is crazy. Some of it's clinical data, but mostly preclinical data. And it's not something that's being used clinically, but it's a representation of how profound this ecosystem, this organ system that we're talking about is, and the role it's playing in all kinds of disease. Another study that was done recently was in autism. And so they gave fecal transplant to a group of kiddos with autism, and they saw really profound improvements. So they gave them FMT repeatedly over 8 weeks. It was a cool study because it was different than what they had been doing previously. And they saw really good outcomes, improvement in both their GI symptoms and their neurocognitive symptoms. And then they came back 2 years later and they had sustained the improvements, and then they sequenced the microbiomes, and those microbiomes still maintained that shift that happened initially with the fecal transplant 2 years later, independent of more treatment, which is also really cool and crazy. So there's a lot of things going on. So there's 322 clinical trials using fecal transplant going on as of yesterday in all these disease groups. And that doesn't mean fecal transplant treats all these diseases, but it means there was enough preclinical evidence to justify a clinical trial to run it in these disease groups. And some of the data is looking really exciting. So what does that mean? That we can use a fecal transplant to look at treating or shifting these disease pathologies. It means that microbes are really important. They're doing a lot of things. They're impacting metabolism. They're making what are called secondary metabolites. The bacteria poop is really important. Our poop is kind of really just bacteria poop in the, really meta way. Let's keep going. Also, they are making things for us that we can't make for ourselves. Like they're conjugating bile acids. It was one of the multiple choice questions. They're making vitamin K. They're processing calories for us. It turns out that germ-free mice eat more and gain less weight because they don't have the bacteria to take that food and convert it to calories for us to use. So they're part of our metabolism. This is my shout out to Lamarck. It's my Lamarck joke. Turns out maybe Lamarck wasn't totally wrong because the bacteria in our gut are evolving based on Darwinian evolution, but they're also impacting our genetic makeup via transcription factors, which was another one of those questions. They're shifting our genes, and they're changing the way that we express our genes through their metabolites. And so Lamarck was the counter to Darwin in the evolutionary debate. And Lamarck said if the giraffe stretches his neck really far, his offspring will have longer necks. And Darwin said, no, you're wrong. Shame on you, Lamarck. It's all random and natural selection. But if the bacteria are evolving the Darwinian way inside of us, but impacting us on a global sense, that might have impact on us in a more short-term epigenetic way that, maybe Lamarck wasn't totally wrong. They impact our immune system. They impact our gut, the gut-brain axis. So they're talking to our brain. They're doing all kinds of cool stuff. How are they doing this? Probably as a hormonal endocrine-like organ. So the 3 pounds in our colon is eating the fiber we eat, eating the mucus layer we have, and their poop, we eat the food, the bacteria eat the leftovers, and then their poop or metabolites is what is acting as this hormone organ communicating with us. And so this chart kind of represents different things that the bacteria make and their function. So they're making the serotonin, part of the serotonin that we use. They're making part of the cortisol. Short-chain fatty acids are really, really important, and I'll talk a little bit about those. They're the main secondary metabolite from fiber, which is why vegetables are so important. And they're the key to communication. So you have all these molecules moving from your colon through your liver into your bloodstream, doing all kinds of stuff. I mentioned the transcription factors at the beginning. The short-chain fatty acid butyrate is something that changes the way our cells function. It rewrites the translation of our DNA, which is really cool, the Warburg effect. It actually turns off cancer cells, colon cancer cells, but it feeds healthy cells, which is crazy. This is a photo taken by the Sonnenbergs. This is one of the reasons I love them so much, their lab. This is that ecosystem. So this is all the bacteria. This is the green, so the bacteria are the speckles. The green layer is the mucus layer that separates the bacteria ecosystem from human cells. The blue is the human part. And it's a really beautiful forest or distant galaxy, however you want to see it. And when it's working well, it keeps us happy and healthy. It makes neurotransmitters for our brain. It keeps us from being sad or anxious. But if that mucus layer goes away, bacteria that would otherwise be fine, it can become bad. And so this is there's a difference between a pathogenic bacteria and an opportunistic bacteria that's maybe making metabolites that are pro-inflammatory, meaning bad for our body. It's not that the bacteria in themselves is bad, it's that their environment is allowing them to do things, just like an invasive species in the forest. If you have too much English ivy, or the English ivy's in the wrong place, it doesn't mean that it was bad when it originally lived in its ecosystem, but in the new ecosystem it can cause all kinds of problems. And our immune system is there to pay attention. So the immune system in our gut is really important, one, because we have this 3 pounds of bacteria that's potentially harmful, but also because it's educating us and we're creating this interface. When that breaks down, we get dysbiosis. Bacteria mediate immune impact through some really interesting ways. I was like, maybe this isn't the best slide in the world, but it's my favorite, and there's somebody out there who's like a toll-like receptor PhD, and they're like really excited right now. My mom was like, don't say it's a bad slide, just tell me about it. And I was like, okay, fine. It's really exciting. This slide is crazy. I don't expect you to read it all right now, but the fact that this origin column tells you about all these bacteria and the digestion of their fibers create things that affect this aspect of our immune system. So for example, digestion, creating short-chain fatty acids like formate, acetate, and butyrate binds GPR41, which affects leptin production and regulation and balance. So leptin is important in obesity. So bacteria are playing a role in the obesity epidemic. We don't know exactly how, but we're starting to tease apart some of these details, and it has to do with these metabolites binding our immune cell and giving them information, changing the way things are working. In my previous life, I worked in bone marrow transplant, and I studied what's called hematopoiesis. And so that's a special kind of stem cell that we used to think was only made in bone marrow. But it turns out those cells are also being made in the gut. What? That's crazy. Well, they're probably interacting with the microbiome if they're down there. And so this study was only published in 2018, but it showed evidence that hematopoietic stem cells are present in our gut. It was a really interesting paper. And honestly, if you guys want this slide deck, just email me at the end and I'll send it to you. Because we had no idea hematopoietic stem cells were being formed and maybe matured, and now we are starting to think about what role is the bacteria playing down there. It's crazy. They're also talking to our brain directly and indirectly. The vagus nerve is my favorite thing to talk about. It's the coolest. It's what controls digestion. And if you think about taking a deep breath, then relaxing, the rest and digest portion of your nervous system, that is the vagus nerve. And these bacteria are speaking to the vagus nerve, and the vagus nerve is talking back to them, which is crazy. They're also creating those metabolites that are going into our bloodstream, which is the circulatory system, circulating throughout our body and talking to the rest of our organs, specifically our brain. Neurotransmitters are being made down there. When you think about that autism study and the impacts you saw on neuroatypical behavior in autism, and think about this gut-brain connection, it starts to make a little more sense why fecal transplant could have such a profound impact on their neurologic stuff that's going on. And then, like I said, immune system. And you start to think about chronic disease and the immune system's role in putting the fire alarm on when it shouldn't be. Maybe the bacteria playing a role in that overhyping of our immune system when it should be just shh, calm down. But the bacteria are saying, I'm not getting enough fiber. Turn on the fire alarm. Neuropod cells were recently discovered. Neuropod is from 2015, and then 2018 they were identified in the human gut. But if you think of like these metabolites we're talking about entering the bloodstream, that's like snail mail. And then all of a sudden we discovered that there's actually an email in there, and we didn't even know it. It's direct neuronal electrical signaling to the brain that's protruding. They're called neuropods. So they're neuronal cells that are like blubbing out into the gut and communicating with the bacteria. So they're talking to the gut, which is really cool. The nervous system is a really important part of this, the autonomic nervous system. I'm going to transition a little bit away from the basic science to the applications of translating this to everyday life, because I think that's really important and probably why some of you guys are here. We have these two sides of the nervous system. And like I said, the vagus nerve is part of the rest and digest or parasympathetic nervous system. And that's on a teeter-totter with the rest of our lives, which is fight or fight, sympathetic. Oh my gosh, the traffic to Vancouver is so crazy. Stressed out side of the nervous system. And the microbiome is playing a role in how this nervous system is teeter-tottering, and how we teeter-totter it with our lifestyle and aspects of daily life also impact the microbiome. So again, take a deep breath and imagine you're in that like very calm, about to eat the most delicious bite or sip the most delicious sip of something really delicious, and feel the salivation in your mouth. That's because the parasympathetic nervous system controls digestion top to bottom. And if we're not in a calm space when we're about to sit down to eat, our digestion isn't even really doing its job. And that impacts the bacteria in the colon and what they end up with and how the food we eat gets digested. I work with a lot of, separate from the fecal transplant, I work with a lot of functional GI issues. And you would be surprised how many people have trouble pooping. It's a really big problem, and a lot of that comes down to us being too far in the sympathetic side, and the microbiome gets angry and then causes this kind of overreactive state. So the solution comes down to the foundations of health. And I love that about the human microbiome. So we talked a lot about like what we're learning about it. We're in the exploration phase. Correlation is not causation, and we have a lot of preclinical data, but we know enough now that the microbiome is a mechanism of action to connect this bridge of all these different aspects of medicine and science, which is really cool. And you have people like the Sonnenbergs coming up with what they called the Big Mac diet, which I thought was really cute. This is from Erica and Justin Sonnenberg, where their joke is microbiota accessible carbohydrates, which is what MAC stands for, is really important. And they're giving nutrition advice. Microbiologists are giving us nutrition advice about how to eat vegetables. And you have neurologists talking to ecologists, which is really cool. And you have naturopaths on stage of Science on Tap, which I love. It's really fun. I want to go out on a limb a little bit and tell you a cool story. There is, and again, just keep in the back of your mind, this is some preclinical data, but I like to think about where this is going to lead us in our future of appreciating the importance of our food systems. And we know that there is a metabolite, so bacteria poop, that is made from carnitine, which comes from red meat, that is associated with cardiovascular disease. It's called TMAO. And it's really hot right now, topic in research. And it has to do with cardiovascular disease, and having high rates of TMAO in your blood puts you at risk for cardiovascular disease, and it's coming from our gut bacteria. And it might have something to do with why high red meat diets have a correlation with cardiovascular disease. But this really cool scientist thought, why don't we add garlic into the diet of these mice we're looking at TMAO, because traditional diets are important. And what role do herbs and spices play in mitigating chronic disease now that we know all this about the microbiome? And it turns out when you added allicin, which is a derivative of garlic, in with the carnitine, which comes from red meat, the mice stopped making TMAO. So if I was going to make a cookbook, which I'm not, but this is my joke cookbook, I would call it the chopping vegetable diet, where really you're just chopping vegetables for 30 minutes a day and using some common sense principles to feed your microbiome. And I come back to this and go full circle, because when I talk about eating poop to save your life, we're a long ways from that being available. There's a lot of barriers to overcome in fecal transplant. I think it's important for people to know about it because it exemplifies the importance of this system. But we don't have to eat poop to start to feel better. We can just start to eat vegetables and think about what's happening in the ecosystem, our vagus nerve, our nervous system. I ask you, what will we learn next? I think it's really important to appreciate where the microbiome brings people together in science. I love microbiome conferences. I invite you guys to email me. The last time I did Science on Tap, I had some great coffees with the head of veterinary medicine at OHSU and the fecal transplant he was doing and his animal models. So feel free to get a hold of me and ask me questions. But really it just comes down to taking a deep breath and eating your vegetables. Thank you so much.
Thank you. Very nice presentation. As we go forward with fecal transplant, is there any vision toward preserving your own diversity so that when you go to the endodontist and they give you that massive dose of antibiotics, you can actually recover? Yeah,
it's called autologous fecal transplant, and it is being done. It's still considered a fecal transplant, so it's still illegal to do without a clinical trial. So it's regulated the same way fecal transplant is, even though it's your own poop, because there's still infectious disease risk. But there's really cool data showing that autologous FMT, so my poop that I collected before I went and got the antibiotics and got C-Diff, will actually still cure the C-Diff. So it is really valuable, and there's lots of people looking into that. It's just not something you can do legally through this conventional drug model yet. But it makes total sense, and people are doing it, and there's good data that shows it can help mitigate infectious disease risk and repopulate you after antibiotics.
Great topic, thank you. I hope you didn't answer this question already, and it just went over my head in your presentation. I'm not understanding how you do the fecal transplant and not contaminate the new fecal that you're putting into the body that has the C-Diff. Does that make sense?
So what is happening with C-Diff is you are out competing, or there's something going on with the bile acids. We're not exactly sure totally what the mechanism is, but as long as the donor poop is screened, the poop is fine, and you're literally swallowing the pellet of it, or it's being delivered via colonoscopy, and it doesn't matter, whatever's there, you're putting compost on the ecosystem that's there to outcompete the invasive species. So it doesn't matter what's the native ecosystem, you're just helping that ecosystem return to health.
So I work with eating disorders, and I'm curious if there's any data on how disordered behaviors such as restriction, purging, may long-term have an impact on the microbiome.
It has a huge impact on the microbiome. And what we have seen with even things, so I also didn't talk much about what is a therapeutic diet called low FODMAPs or orthorexia, which is something we see a lot, which is disordered eating where people are sick and lots of foods make them sick, and so they really limit their food to no fiber. And one of the theories is that those people don't have the guild that can process that fiber, but not having the fiber also starves whatever you have left. And so there is data showing, sequence data showing when you are on a restrictive diet or in anorexia or even bulimia and purging, that it shifts the microbiome towards an unhealthy ecosystem. But so does a standard American diet. And so does, a diet that has low fiber. And you can't just overnight go to a high fiber diet. Just like any ecosystem, you have to adjust. So fiber has to be introduced really slowly back in. I hope that kind of answers. But yes, it absolutely impacts the microbiome.
Hi. I make a spinach smoothie every morning for my older teenage boys, and they just love them.
Good for you.
They hate them. I force them to take it because I know it might be their only vegetable of the day, right?
Yeah.
So my question is, I'm putting that in a blender. I'm putting kale and I'm putting spinach in a blender with other nice things that are, that have some prebiotic and stuff. But if I blend it, do they get the same gut
value?
Stuff? Yes.
Yeah, and I think this is a good place to talk about the difference between a smoothie and juicing. Because a smoothie, you're maintaining the fiber, which is great. And juicing, you're getting a lot of the good plant polyphenols, but you're not getting the fiber. And so it's not that juicing is bad, but smoothies are better from the perspective of you want to get that fiber, in addition to all the good stuff that comes from the spinach and the vegetables and all those things. And it's not that juicing is bad, it's just that you're not getting the fiber when you juice, but you are getting it. And honestly, my favorite thing about the microbiome is I explain it to people and myself, and it makes it way easier to do the dietary changes because they understand why it matters. So maybe give them there's an NPR video about the microbiome, that's great.
Hi. So I think I heard something about with the transfer FMT where they tried removing the bacteria and filtering even the bacteria out, and it was still effective.
Yeah,
Can you talk a little bit more about that?
So we don't know why fecal transplant is so effective in C-Diff, and when they freeze dry it, it works. When they freeze it, it works. When they leave it fresh, it works. And they even filtered out the bacteria, and it still works. It's a sterile filtrate. So that's why I talk a lot about those secondary metabolites and how these cells are communicating with each other. I have a very heated debate with the virus people because the bacteria filter takes out the bacteria. It doesn't take out the viruses. So it could be the virus, the good viruses that are there, but it also could be all these metabolites that are shifting the ecosystem more analogous to like a compost than literally replanting plants, if that makes sense.
Hi there. I'm a dietitian. I work with people every day who I feel like have gut problems, and they may not agree with me. But one of the things that you said in your talk really bothered me, and that was when you lose a species in the microbiome, you lose the ability to digest certain types of fiber. And so I've really struggled with going the route of like low fiber foods and then trying to reintroduce fiber foods that people can tolerate. But I have limited ability to like test the microbiome and see what species are alive in people's guts. So how do you know like if you're prescribing the right things? Like if I'm telling people, like I tell people like eat more fiber every day, all day, like eat more beans, eat more beans, eat more beans. But what if they don't have a species to digest those foods?
Yeah, and that is where this gets really complicated because everybody's microbiome is different. And we are in that exploration phase, so we don't have good testing. I don't do functional testing to see what kind of species there are because it doesn't really mean anything yet or doesn't mean a lot yet. It means some things, but not a ton. And so it really comes down to helping people listen to their bodies and testing food slowly, but a big asterisk to that is their nervous system matters. And so they could be eating all the right things one day and the next day be preparing for a test, and they're not going to digest any of that. And so it's a very delicate balance of the chicken or the egg. Is it because you're in sympathetic mode and you're not digesting anything? Is it because you've lost the guild that can process inulin and that's just going to make you super sick? So it's a trial and error, and if I had the right answer, I would publish it on the internet, and it would be great, but it ends up being an hour-and-a-half conversation with patients instead, where we make a plan that's much more specific and individualized. There are definitely people who respond better to low-fiber diets, and it's a saving grace to them, and it's like the first time they felt better, and the question is, how do you reintroduce the good guys? And Martin Blazer and Maria Dominguez Bello have some opinions about that, and the role antibiotics play and our loss of ecosystem diversity. And fecal transplant might be part of the answer in the future, but we're not there yet.
How much data do we have on using fecal transplants as a treatment for IBD, irritable bowel disease?
There's a reasonable body of clinical research for that. I didn't mention it also intentionally because it's mixed results, and so it's not clear-cut. There are a handful of Crohn's and ulcerative colitis, which are the two kinds of IBD, studies that have shown it does induce remission in some people. It doesn't in everybody, and it can cause flares. And so sometimes it makes people worse than they were, and sometimes it makes people better, but it's not clear yet exactly what is going on. And IBD as a disease is also really complicated. And so when we look at Crohn's and colitis, there's not even uniformity in what their microbial profiles look like or what the underlying mechanism is. And so that probably has something to do with why fecal transplant doesn't seem to do the same thing in everybody.
All right, thank you so much, everyone, and let's give it up for Andrea McBeth, ND.
Thank you.
Thanks for listening. If you're in Portland and want to come to an event, check out our website for a list of upcoming topics at scienceontaporwa.org, and that last part stands for Oregon and Washington. There's more about our events on Facebook, Twitter, and Instagram at scienceontaporwa. As usual, I want to say thanks to my awesome group of volunteers, including Scott Fry, Chris Gowan, Sam Lauk, Rita Nygren, Steve Perry, and Chris Trone. These events and podcasts would not happen without them. A special thanks to Amber Peoples for hosting this event and to Graham Tully for audio production. Also, a big thank you to Jonathan Coulton for letting us use his song Mandelbrot Set as our intro and outro music.
Just in time to save the day.